Welcome to the Student Spaceflight Experiments Program presentation. We are very excited to be here. Our project is titled The Development of Venessa cardui butterflies in Microgravity. We are from Kent Place School, an all-girls independent school located in Summit, New Jersey. Our team was made up of 5th and 7th grade students. Our co-principal investigators are Olivia, who cannot be here, myself, Alexandra, Isabella, Nora, Aya, Abigail, and Elizabeth. Our teachers are Mrs. Crow, our 7th grade science teacher, and Mrs. Van Rye, our 5th grade science teacher.
Our purpose is to examine the role of microgravity on the growth and life cycle of the Venessa cardui butterfly. We would like to examine the role of microgravity on their development from egg to chrysalis. Microgravity results in the feeling of weightlessness on the ISS as it is in free fall around the Earth. On the ISS, the eggs will need to find a way to hatch, and then the larvae a way to suspend, in order to form the chrysalis and, in theory, emerge and expand their wings. Due to the fact that this species of butterfly needs to suspend upside down in order to pupate correctly, the role of microgravity is essential. Our justifications of the purpose of this experiment are twofold: to examine the viability of eggs in long-term cold dormancy in order to be transported to space and possibly support sustainable insect populations, and the possible long-term benefits of pollination assistance. Butterflies are pollinators, and the pollination process leads to healthy seed and food production of plants. The butterfly's assistance to pollination can lead to easier food growth, a crucial component of future long-term space travel.
The butterfly being used for this experiment is Venessa cardui, also known as the painted lady butterfly. Painted lady butterflies can be found in almost every country in the world, making them a well-known species to experiment with. Venessa cardui have a wingspan ranging from 5 to 9 centimeters. All butterflies start out as eggs, which are about 1.5 millimeters, the size of a pinhead, and they normally hatch after about 3 to 5 days. After the larvae hatch from the egg, they feed voraciously as they continue to grow as caterpillars for about 7 days, while getting ready to start the metamorphosis process. The larvae need to find a suitable place to securely attach themselves and hang upside down in order to form a J shape, which they then shed their final layer, which allows them to form a chrysalis. While inside the chrysalis, the butterfly turns into a liquid mass by releasing enzymes that digest the caterpillar cells, except for the essential imaginal discs, which then develop into adult butterfly structures. This allows the larva to rearrange itself in order to become a butterfly. The butterfly emerges from the chrysalis after about 7 days, which ends a 2-week development process.
In order to have the eggs arrive in a dormant stage on the International Space Station, the team did some ground-proof tests to check the ability to keep Venessa cardui eggs dormant using cold storage. Our first experiment involved the refrigeration of the eggs for 4 days, causing the larvae hatching process to be successfully delayed by 3 days. The dormancy process is necessary because if the eggs are refrigerated prior to the spaceflight, they will remain dormant, causing the life cycle process we wish to experiment with to only occur while in microgravity. Our second experiment increased the days of refrigeration to be more aligned with the actual travel process to the International Space Station. We placed approximately 35 eggs in the refrigerator in early June. 1 week after refrigeration, we took 6 eggs out from the cool environment, placed them in 3 separate small containers at room temperature, and watched to see if the growth process would be successful. 50% of these eggs successfully hatched and are currently in the larval phase. Additionally, after 20 days of refrigeration, none of the refrigerated eggs had hatched, and we removed another 3 samples to test for normal growth and development.
Setup of the FME tube is essential to the experiment. The first part of the tube, volume 1, will have 5.0 grams of culture medium larva food for the larvae when they hatch. The second part of the tube, volume 2, will have 4 Venessa cardui eggs. The tubes will have been refrigerated to keep them dormant until they reach a space station. The reason why we have 4 eggs is to ensure the survival of some eggs. The third part of the tube, volume 3, will have 3.5 milliliters of fixative. This solution will function to stop their growth process prior to leaving the ISS. It will also preserve them until we can observe them at our school. Our procedure states that on the 3rd day after the arrival on the ISS, the astronauts will unclamp clamp A. This will allow the hatched larvae and the food to be in contact with each other. Based on our experiments, we have decided to open clamp B to release the fixative 5 days before undocking to allow the larvae to reach the chrysalis stage, and also end our experiment before it leaves microgravity.
To review, our experiment proposed to send 4 Venessa cardui eggs in microgravity in order to observe how the environment affects the hatching and development of pupating insects, as pollinating insects are a vital part of the viable ecosystem. Our hypothesis is that the cold storage will successfully delay the hatching of the eggs and allow for normal development of larvae in microgravity. They will then form a chrysalis in space because they can attach to something, making their development in microgravity possible. When our experiment returns from space, we will collect our post-flight data by counting the number of eggs that hatch, how many larvae pupate, and what stage of development they obtain before being fixed. We predict that this study will have a positive impact on future experiments and space missions because, as adults, butterflies pollinate flowers and gardens, producing fresh food for the astronauts and scientists in space, and making it easier to have longer and more advanced flights in space and microgravity.
Thank you so much to the SSEP for the opportunity to present our flight experiment. We would like to acknowledge Kent Place School and our science department faculty for supporting us in our research and experience, Dr. Meg Ballard of the University of Delaware for Skyping with us and teaching us about butterfly development and helping make our project successful, and Verizon for their financial support of our project.
Do we have any questions for this team?
Hi. What made you all choose that specific butterfly?
So we were originally going to use a butterfly called the Western Pygmy Blue Butterfly. It's the smallest butterfly in the world. It is the wingspan is 1.5 centimeters, about half the size of your fingernail. But due to the rareness of the Western Pygmy Blue Butterfly, we were unable to find it. So we went to the Painted Lady Butterfly, which is a very common butterfly. And even though they are too big to fit in the tube at their full-grown stage, due to the frozen eggs, they will be delayed in their hatching. So they'll likely only get up to the chrysalis stage, and that will be able to stay in the tube. And then they will be killed by the fixative.
Thank you.
We also chose it because painted ladies are known because they are found like anywhere, and they're easy to adapt to different habitats, so it would be good for this experiment.
Okay, thank you.